Hi everyone, colleagues and fellows. My name is Dong-Soo Kang, and I will take today's webinar session with the title of Expedite Your Success, Risk Mitigated Biosafety Testing. Starting from self-introduction, I'm in charge of Biosafety Testing Services, aka BTS, in CRO business team at Samsung Biologics since February 2019. Before joining Samsung Biologics, I went through site facility manager at WuXi Biologics Biosafety Testing, head of viral clearance at WuXi Apotec Testing Technology, both located at Suzhou, China, and viral vector manufacturing at WuXi Advanced Therapy located in Philadelphia, US. Educational background is protein biochemistry and molecular pharmacology focused on G protein coupled receptor. Today's presentation will follow as in order of biosafety testing overview, lot release testing, viral clearance validation, and key takeaway as lesson learned from a field experience. Modern day medicine and drug provide a great benefit to struggling and fighting patients with a wide range of disease. By controlling symptoms, curing disease, and even protecting us from potential one. Since it is used for human body, safety is the priority concern. As we all know, the patient safety always comes first. Because of that, health authority require thorough biosafety testing during manufacturing process. This is a simplified diagram for mammalian cell-based recombinant protein production, such as monoclonal antibody, fusion protein, etc. Multi-layered safety barrier positioned at critical step and in between. From the beginning, extensive testing is required for master cell bank and working cell bank testing as cell line characterization. Bulk harvest testing utilized worst case sample for upstream process. Viral clearance study validate downstream process, not only purify target molecule, but also eliminate adventitious agent which could contaminate during manufacturing process internally or externally. In order to release drug substance, there is a set of biosafety testing to assure the product safety. Major business of Samsung Biologics is contract manufacturing, which we manufacture variety of drug substance for broad range of clients worldwide. That's why we set up the lot release testing first among other biosafety testing as a CRO business. Lot release testing consists of mycoplasma test, adventitious virus test, which is so-called in vitro assay, minute virus of mouse, MVM or MMV detection by quantitative PCR, and traditional microbial tests such as serology bioburden and endotoxin test. To perform such a testing, dedicated facility and critical materials are needed. At BTS, we are prepared by a series of mycoplasma positive control for culture methods such as Mycoplasma herinis, oreille, pneumonia, gallisepticums, Leigh-Brawy, and palmentans. And indicator cell lines and virus positive control for in vitro assays such as MBM, parainfluenza virus type 3, type 5, herpes virus, and measles virus. Based on our experience as a client for biosafety testing, once in a while, we found a few risk factors we might consider, such as the presence of a strong quality organization, potential issue during shipping and handling, as well as time for logistics, and solid program of sample management, and extension of turnaround time by retest. I will go through the possible solution as a risk mitigated approach using BTS as a showcase. Based on our experience as a client for biosafety testing once in a while, we found a few risk factors we might consider, such as presence of a strong quality organization and potential issue during shipping and handling, as well as time for logistics, and solid program of sample management and extension of turnaround time by retest. I will go through possible solution as a risk mitigated approach using BTS as a showcase. Here, BTS and Samsung Biologics share CMO QA group and take advantage of their expertise with proven record of solid audit inspection history. For example, last year, Samsung Biologics record 53 accumulated IND and BLA approval from health authorities worldwide, on top of multi-year contract commercialization programs. Not to mention, BTS lab itself passed FDA GMP surveillance inspection together with the CMO manufacturing facility at the end of last year. From the upper diagram, this is more applied to Samsung Biologics CMO and local client. Once samples are generated, testing samples or testing article go through custom and shipped to global CRO companies located at either US or Europe. Then sample will be registered and sometimes redistributed to the actual testing site or testing facility in order to initiate testing. In case of using BTS, testing sample is collected and registered at manufacturing site and released from 24 hours, 7 days employee stationed dedicated sample management group and transferred to BTS, which is located on same campus on a day of testing. This will minimize any potential risk and time for sample logistics. This is a brief overview of timeline for mycoplasma test and in vitro assay, which take 28 days. In general, once test is completed, if there is any issue due to cytotoxicity by testing sample or product, then independent cytotoxicity test is performed on indicator cells used on both mycoplasma test, either mycoplasma test or the in vitro assay. Using dilution factor from cytotoxicity test, retest will be initiated with diluted sample, which take another 28 days. In BTS, our test method incorporate cytotoxicity assay at the beginning of product-specific qualification, which we call method suitability test at Samsung Biologics. Once Once dilution factor is determined, actual test is performed by dual dilution factors. For example, dilution factor is undiluted, then test is set up with both undiluted sample and twofold diluted sample at the same time. Final result will be obtained from a test used less diluted sample as a worst case. This double barrier system as incorporation of cytotoxicity assay and dual dilution factor accommodated test method will make sure there is no issue by product cytotoxicity and will keep promised turnaround time to our client. Viral clearance validation. This slide is generously shared by Dr. Kurt Brosson. Electron microscope image of CHO cell shows retrovirus-like particle displayed as a black circle. In mammalian cell, retrovirus-like particle can reach up to 10 to 9 particles per ml in unprocessed bulk. This can be one of the clear reasons why viral clearance validation is required for downstream process, specifically mammalian cell-based production. To perform viral clearance validation, large manufacturing process need to be scaled down using critical process parameters into lab scale. On the other expression, miniaturized manufacturing process is placed on our lab bench or tables. Each downstream process is validated as how the process efficiently remove or inactivated mother viruses. In case of chromatographic process, no number of mother virus is spiked in input material or starting material, and virus is quantitated from output material after process. The difference between two numbers in terms of virus is expressed in log scale as log reduction value, LRV, which represent viral clearance for the specific process. For more direct example, 10 to 6 virus is spiked in loading material for CEX chromatography on a diagram, and 10 to 2 virus is detected in elevate fraction, then log reduction value is 4. As another example of virus filter that physically removed the adventitious agent by pore size, 10 to 6 virus is spiked to load material and no virus was detected in filtrate, then log reduction value will be 6. These examples are very principled and schematic. Actual log reduction value calculation incorporates statistics such as limit of detection, 95 confidence level, and also volume of tested sample, dilution factor, etc. From electron microscope image, now we know retrovirus-like particle can be maximum 10 to 9 particles per ml of unprocessed bulk sample. If one dose is made up from 500 ml of unprocessed bulk sample, we can consider 1 liter for 1 dose as worst case. Then theoretically, 10 to 12 retrovirus-like particle can be present in one dose. On top of this number, by adding 10 to 3 particles as a safety factor, final number 10 to 15 will be the target log reduction value to be achieved during viral clearance validation. How we can achieve this target log reduction value is a matter of how study design for viral clearance validation. To wrap up, the key takeaway as a lesson and learn: the communication and coordination between testing site as a CRO, client, and vendor as a filter manufacturer, specifically virus filter manufacturer, is important to read planning ahead of actual viral clearance study. Understanding product and process and robustness is always beneficial to successful viral clearance validation, such as critical process parameter required mock run without virus spike, specifically for virus filtration study, artifacts caused by freeze-thaw of the testing sample, product reproducibility, and so on. Before spiking study, toxicity assay, interference assay is a must to determine dilution factor for virus quantitation to make log reduction value. Pure and higher titer of mother virus stock is critical because more virus could bring higher log reduction value in general, except one case. In filtration study, high amount of virus spiking could cause clogging membrane and breakthrough, which will reduce reproducibility of viral clearance study and log reduction value on this step. By testing more samples, limit of detection is getting lower and log reduction value will be increased on same process. The last one, risk is never zero. Viral clearance as well as other biosafety testing is approached by the worst case scenario as risk mitigation. As a CRO services at Samsung Biology, we can provide. We are providing lot release testing service as a part of biosafety testing services in BTS and viral clearance and master cell bank, working cell bank, and storage and the product cell line. We will provide these service at first Q 2022. Currently, we are providing service contract QC and traditional microbial tests such as sterility, endotoxin, and bioburden. Q&A session. How biosafety strategy lead to client win in comparison to global competitors? So in this case, how beneficial to our client in our company? Might say biosafety testing is a mandatory testing required by regulatory authorities. Therefore, we can consider the biosafety testing is a supply chain. We can consider biosafety testing is a part of supply chain in terms of contract manufacturing. When we consider this one is a centralized system, then it will be definitely beneficial to our client because test result will be delivered on time. Effort and finance invested drug substance need to be released by the time. So I see that the biosafety testing at Samsung Biologics will be beneficial to our client at Samsung Biologics, as well as a client located in Korea or elsewhere, Asian Pacific region. Next question, describe present requirement for biosafety testing in current environment. So this might be that actually this is a very excellent question considering that biosafety testing, locational biosafety testing facility in the manufacturing facilities. So this is our biggest concern when we build up the biosafety testing lab in Samsung Biologics. So all about segregation. First, BTS, Biosafety Testing Lab, is isolated building from the other manufacturing site at Samsung Biology campus, which means we have a physical segregation. And also there is a segregation policy by facility-wise and the procedure-wise, which means SOP-wise. All those to avoid cross-contamination. All these effort is a part of our business continuity plan. Let me list the few, then like a separated personal material waste flow, unidirectional, and double decontamination policy, triple gowning procedure, like a PPE, segregated biosafety cabinet and incubator for each sample. For example, negative control, testing sample, and positive control, and so on. I hope that this one, my answer will give that the brief answer for the how we put the effort on segregation between manufacturing CMO and biosafety testing facility. The next question, advanced BSL-2 requirement. As long as I know, there is no, the requirement is more like a general, the personal protective equipment and wearing the glove, more like environmental safety and scientist, like employee safety issue. Also, there is the cross-contamination from BSL-2 lab. Actually, BSL-2 lab, the BSL-2 lab is not categorized as a high or dangerous lab. So, however, it is recommended a single pass HVAC system means like regulated air circulation, less amount of air will be circulated, and also biosafety cabinet, which is B2 type of biosafety cabinet, which most of the air is sucked into HVAC system instead of continuous circulation inside of BSC. And like waste material, control of waste material and regulation of that hazard material. Those things is like some of those I mentioned will be that in a list for BSL-2 requirement. But of course, it's a detail and exact list, total list will be easily find on the internet or any guidance. The last one for today's, how to design viral clearance study. Everyone's manufacturing process, specifically downstream process, is slightly different. So this one is a design. Design will be slightly different too, because it miniaturized the manufacturing process onto the lab scale. That's why you have to use a different critical process parameter. But main outline is simple. For the Western countries, regulatory agency, if you want to file in IND, then you can go through low pH or solvent detergent inactivation, virus removal filter, and chromatography step will be validated. In case of IND, you only use two types of virus, MBM as a worst case scenario because it's because of size and at the high resistant, and XMURB for the mother virus for retrovirus like a particle we just saw in the electron microscope image. In case of BLA, you have to use all the 4 mother viruses most of CRO company can provide. So 2 viruses, 2 envelope viruses for virus inactivation, and all 4 viruses for filtration and chromatographic step. On the other hand, in China, the bioclearance study design will be slightly different because there is no chromatographic process is required. You have to do low pH inactivation and virus removal filter by all 4 virus, except low pH inactivation. In case of inactive virus inactivation, it's only 2. And mother virus is usually commonly used mother virus is XMURB, like I mentioned, is a mother virus for retrovirus like a particle. MBM, this is a worst case virus, mother virus, because it's very small, 20 nanometer diameter. And it has a very high physical chemical resistance characteristics. And PRB as herpes virus, envelope the herpes virus. Herpes virus is another envelope mother virus with double-stranded DNA. Rare 3 also very commonly used. Those four mother viruses commonly used in the viral clearance study is very carefully selected by genome size and genome characteristics and size of the virus, and whether it is envelope or non-envelope, or their characteristics of physical resistance. And also can be found on a regulatory guidance. I think that's all for today's webinar session. Thank you.